Hey everyone, my name is Christopher Cotter, and today I'll be talking to you about my project evaluating the antioxidant properties of phytochemical extracts from an ancient grain teff in THP1 monocytes. Eragrostis teff is a cereal crop grown primarily in Ethiopia that produces the smallest grain in the world. Teff is a desirable crop because it's resilient to abiotic stresses such as drought and waterlogging. It's more resistant to insects than other cereals, it's gluten-free, and it's quite nutritious, with lots of macronutrients and high levels of amino acids, minerals, and water-soluble vitamins. Teff is produced in two main varieties, ivory and brown, with ivory being the more popular of the two and therefore costing 55% more, despite brown teff being more nutritious, containing more calcium, more iron, more flavonoids, and more phenolic acids. Several studies have investigated the phenolic profiles of teff and their antioxidant activity in cell-free small molecule assays such as FRAP, DPPH, and ABTS assays. These studies suggest that teff may be a good source of antioxidants based on their phenolic profiles. However, few studies have investigated other bioactive molecules, and none have performed antioxidant assays using cell models. Glutathione is a tripeptide found in high intracellular concentrations that functions as an important antioxidant and cofactor for other antioxidant enzymes. Decreased glutathione levels have been associated with neurodegenerative disorders, pulmonary diseases, cardiovascular diseases, and other chronic conditions. This study seeks to determine the presence of bioactive compounds in brown and ivory teff by extracting phytochemicals and evaluating their effects on intracellular glutathione concentrations. For this experiment, we used a methanol, chloroform, and hexanes-based extraction process that removes any compounds that are too polar, like sugars, or too hydrophobic, like lipids. The extracts were then separated on a column into fractions, and both the whole extract and the crude fractions were used in the cell treatments. THP1 monocytes are a human cell line used as a model for this study. Cells were incubated with the extract for 24 hours, then the protein was collected and intracellular glutathione content was measured using a fluorometric method. Here are the results from our cell treatments. Figure 1 shows the relative cellular glutathione content after exposure with the brown teff extract. All of the data is relative to the control untreated cells, shown here as V, or vehicle. SM denotes the whole extract, or the starting material, and the rest of the samples are from the extract fractions. Fraction 4 showed the greatest increase in glutathione content here, with approximately a 100% increase compared to the control. Figure 2 shows the relative cellular glutathione content after ivory teff extract exposure. Here, all of the fractions and the starting material show a small increase in the glutathione content. However, the starting material and fraction 4 both show about a 50% increase in the cellular glutathione content. Since we previously found that fraction 4 from both brown and ivory extracts showed the most activity, we decided to test the brown and ivory starting material and fraction 4 head-to-head in the same experiments. Here in figure 3, you can see that the brown teff fraction 4 had approximately a 190% increase in glutathione content and was significantly different from the control and all of the other treatments with a p-value of less than 0.01. Brown starting material and ivory fraction 4 were also significantly different from the control and the ivory starting material, but showed a much smaller increase in glutathione content. In conclusion, bioactive crude fractions were found in both brown and ivory teff. Brown fraction 4 had the largest increase in glutathione content with a 190% increase compared to the control. Moving forward, we were planning to identify the compounds in the active fractions with the help of the Oberle's group. We're also planning to do cell viability studies and microscopy, and finally we're planning on doing qPCR studies for gene expression. Thank you for listening, and feel free to contact me with any questions.